So Sugu, are you here? Yeah. Yes, I think Sugu is here. Yeah. So thank you, Sugu. So please start sharing the screen, Sugu. I'll probably stop here. So friends, please allow me to take honor in introducing Sugunakar Vure, my good friend who is an assistant professor based in a lovely professional university. He obtained his PhD from Osmania University in Department of Genetics, coming from a wonderful PhD on acute myeloid leukemia, which is a well-known blood cancer. And by the time he finished his PhD, he has gotten an amazing number of 14 publications. Okay, which is quite phenomenal, which probably no PhD fellow would have ever gotten it. And after that, he has obtained almost another 30 publications, where he went on to do his postdoc in Korea. And he was also an ICMR postdoctoral fellow. And most importantly, he has been a wonderful helper and a mentor to as many as n number of students across India, and more precisely for Bioclues community. He's also owned a prestigious position in Bioclues as a secretary, and he has really been very much helpful taking the Bioclues baton to the next generation. So we'll talk until 6:40, and then you will just hand over to me, where you will demonstrate cancer genomics. So over to you, Subbu. Thank you so much for your time. Yeah, please.
Yeah, Prash, thank you so much for the wonderful introduction. I'm glad, yeah, to be part of this workshop. I hope I'm audible to you, to everyone. Prash.
Yes, Sugu. And
Yeah.
Sugu, I'm just logging off from here, and I'll again join it exactly 7:35. Thank you.
Yeah, sure, Prash. Thank you so much. Yeah. Yeah, hello one and all. Good evening. Uh, I'm happy to be part of this workshop, talking to you about something on cancer genomics, to the little bit what I know, learned as a part of the journey from a PhD to till now. Uh, yeah, I don't need to give for the introduction. Yeah, I have, when we try and talk about cancer genomics, we always get one question that in mind, everyone over here. For example, I'm basically from cancer genomics or cancer genetics. So, do you know that we are increasingly able to detect cancers by just testing just a blood sample, or that we're moving towards treating cancers not by where they are found in the body, but how their genomes have changed. Uh, I think most of you have understood during this particular workshop. Now, when trying to understand what exactly cancer, genomics of cancer is all about, I don't need to, who have worked in cancer, no need to tell you, but I might, I'm not
Uh,
sure. Hello.
so it's Jayashree here.
Yes.
Maybe I'm not hearing properly. I don't know. Little.
Is it the same with other people or only with Jayashree?
Now you are audible, but before some time.
Close, it's better.
It's better.
Yeah.
Anshumam, it is good.
Yeah. If you leave the mic down, we're not able to hear.
Okay. Yeah. Yeah. Fine. Yeah. I'll try to hold it. Yeah. Now, as we talk about cancer, it is very well known that it is a heterogeneous genetic disease that results from changes in the genome of cells in the body, which leads to uncontrollable growth and which involves sort of genetic mutations that happens in the genome. Um, and our cells constantly uh, finding and fixing those mutations with the repair system that what the body contains that occurs in a genome, and then as the cell divides over and over again, so the repair system obviously takes place in the body. That's how the cancerous cells or the mutations that what we see, they'll be eliminated from the body in general cases. Okay, because every day we are exposed to various agents, but on rare occasions some mutations slip our cells, because when we still see the population in general, we don't see that all the people are with cancers. Yes, their body system is well protected by their system, and sometimes it gets slips, and the repair machinery and those repair machinery also get missed, and those mutations which lead to cancer. Now, when you try to understand the Human Genome Project, which try to establish what normally usually looks like the human genome, now we come to know that what exactly the human genome is all about. Once we know that what is a human genome, then we try to know what are the genes which are responsible for what kind of function in a normal sense. Okay, when you try to understand, when you go for sequencing, then you understand how the genes are getting mutated and which is leading to cancer. Now, when we try to see large projects around the world which basically undergo, the most important thing is here the TCGA database, the Cancer Genome Atlas, which is in US, and then the Cosmic database, which talks about mostly about the catalog of somatic mutations in cancer, which is from United Kingdom, has determined the genome sequences of thousands of cancer samples. It's more like a repositories where you get to download those things by taking some permissions, and then you log in. Then you can able to get the data, and that's where you try and understand about those uh, uh, cancer samples. Then you start working on them. Now, when we try and see, we already discussed like cancer is caused by changes in your genome. That's where like yesterday we have seen this Sarkos image by Harpreet, who tried to show you like how exactly to build by using Galaxy. I think you are quite aware of this, and this is a part of a Sarkos image from acute myeloid leukemia, where you try to see different kind of combinations with the fusions, genetic rearrangements, and so on and so forth. Mm, So it's important with the advancing technologies with regard to NGS or single cell genomics currently what people are using, a new understanding of cancer and new ways for diagnosing and treating many types of cancers have come into into the forefront now. Now, when I try and understand a comprehensive analysis of acute myeloid leukemia, for example, I'm just taking an example here. We try and see that there is something called as Gemlin system. Then you have a somatic system. Okay, and when you try to take about a skin cell, which is also called as melanoma, basically you see there are a lot of germinal mutations that basically prevails. But whereas AML, by virtue of its uh, development, it's basically somatic in nature, which is sporadic, which is induced by the environmental factors. Uh, so when we try and understand this particular uh, the combination between them taking skin cell as a control and then AML blast, there are many features that one can study as a part of the development. You go for gene expression profiling, you go for RNA sequencing, RNA-seq, including microRNA technology, microRNAs. Then you do methylation analysis where you do epigenetic aspects. So you study different kind of approaches to understand how that particular how the epigenetic and genetic factors are influencing in the predisposing of the disease that is taken into consideration. Okay, now when you try and understand when I try and understand the chromosomes, I think someone can you just mute your mics? That'll be nice. Yeah, yeah, thank you. Then when we try and understand about the genetic and epigenetic aspects into consideration, so based upon the kind of analysis that what we do, it has been divided into tier 4. We try to see the mutations, we see repetitive elements, we see non-complex regions. Within the chromosome studies, we see copy number alterations, uh, uh, deletions that we come across. And so there are a lot of kind of, you see a number of mutations that basically takes place, the amino acid coding regions, annotated exons, and pre-mRNA splice regions, and so on and so forth. When we talk with regarding to AML taken into consideration, for instance, presence of DNMT3A and NPM1, uh, nucleophosmin P1, and MLL mutations that influence dose response to daunorubicin, and which is to treat AML. Now, what exactly this particular DNMT3, NPM1 on MLL? The presence of mutation, the presence and absence of mutation is one phenomena that one need to understand. For example, you see that there are different combinations of mutations that basically arise. I'll just quote one example with regard to FLT3-ITD. Now, with regard to AML, there is one important gene called as FLT3-ITD. It is an important prognostic marker. I'll show you in my slides. It's part of my study I did. FLT3-ITD is an oncogenic mutation by its nature. It's basically a tyrosine kinase molecule, which is a part of cytokine signaling. Presence of FLT3. It's a general FLT3. It just helps in normal tyrosine kinase production, and it does it does normal signaling pathway. But because of mutation in this FLT3-ITD, it so happens that the ligand which has to bind onto the membrane, okay, without it's called as ligand independent dimerization, where because of the mutation in the particular receptor, in the receptor, it undergoes constitutive expression. Even there is no binding of ligand, still it's undergo mutation. It's called as ITD, internal tandem duplication. This is a mutation that forms in that particular uh, patient per se. Okay, and presence of this particular mutation will undergo constitutive activation, and you see the abnormal production of this particular protein. Okay, that's where it creates a problem to the normal cellular environment. Now, in the study, what we have done in my study, like for example, I have done around 226 samples were taken into consideration, and we have tried to screen in Indian population. We could able to see 17%. For example, out of 226 samples what I've taken, 17% of people, the patients who are come with AML, they're having this particular mutation. What exactly happens if this mutation is present? They don't respond to chemotherapy. They don't respond to chemotherapy, even though the regimen is given 7+3 daunorubicin, and the regimen is given. They don't respond. My intention is that unless until you understand the genes, that the oncogenic mutations, then when you understand the tumor suppressor genes, when you don't understand the low penetrated genes, by doing this kind of analysis, you will not come to know about how to solve the problem. Okay, so as yeah, there are a lot of things to go. I'll try to rush it up, and this is one kind of analysis, like with regard to AML. When you're trying to see the favorable side, so there are many factors, and AML is associated with genetic abnormalities too, where you see chromosome abnormalities, M0 to M7, different kind of translocation that is takes place. Yesterday, Dr. Harpit was talking about something called as 922 translocation in CML patients. It is chronic myeloid leukemia. I was talking about that. It is 922, where you is try to show that fusion genes, how it going to takes place. How can you do it by exomic, by using Sarkos analysis. So that's how you're able to understand those kind of things. It's all about translocations. Now in AML, it contains M0 to M7 translocations, different kind of translocations that is there. So presence of abnormal cytogenetics, it's about of unfavorable cytogenetics, intermediate cytogenetics, and favorable cytogenetics. Under these conditions, what are the genes which are part of it? That's where the complexity of the cancer come into picture. Any cancer is forming, it is because of different factors. It's not a one factor is responsible. That's the reason we call it as heterogeneous in nature. The culmination of oncogenes, tumor suppressor genes, and the low penetrating genes, which are called as SNPs, which will help in predisposing in the development of cancer. It is not one factor which is responsible. We talk about tumor suppressor genes, we say it as loss of function mutation. Please understand, tumor suppressor genes, it's a loss of function mutation. When you see oncogenic mutation, for example, FLT3-ITD, okay, nucleophosphamide is also an oncogenic mutation. CBP alpha is also oncogenic mutations. You are able to see this TET, ALXL1, IDH1, okay, RUNX1, FLT3, NPM1. These are most of them are oncogenic mutations, and you have something called as DNMT3, which is an tumor suppressor genes. See, there's a lot of complexity that basically takes place. Then you talk about PML RARA, the fusion genes, and RUNXT1 and MYH1 CBP. So there is a different combinations of genes that basically happens at a given condition in the particular patient. Now, most important thing you can ask me. So like we always talk about genetic stability. What is genetic stability? It is one of the it's the most strictly retained by cell surveillance mechanism. Every individual has got a cell surveillance mechanism, which will safeguard the cells from damage and avoid abnormal cells being propagated. That's what called as cell surveillance mechanism that every individual body possesses. Okay, and these mechanisms mainly include cell cycle checkpoints, DNA repair, and apoptosis. And the problem comes when the catastrophe of self-reliance mechanism is failed, or it is with survival of mutated cells, and this leads to propagation. This is an important point of understanding what factors will influence how the cell surveillance mechanism is going on. So it's a point of discussion. Yeah. Now, when we try and understand about this clinical front, taken into consideration about this complex disease variants, Mendelian disease variants, cancer germline mutations, I'll be most focusing on cancer somatic mutations, and that's where I'll try to show regarding cosmic database. I'll just try to give you some glimpse on it. I'm just trying to show that particular thing. So. So when you try and see these functional annotations of four types of disease-associated variants, uh, uh, that is that has been seen, uh, the consequences by Mendelian disease variants, nothing but the rare genetic variants that we see into consideration. Cancer predisposing germline variants that basically we see in some kind of germline mutations with regard to different cancers, for example, breast, pancreatic, and then prostate, and those kind of cancers. And recurrent cancer somatic mutations are more serious than that of complex variants. So we need to understand those particular variants to have a clear-cut idea. Now, when I talk about SNPs, I'll be talking about most SNPs and then oncogenic mutations, a different kind of analysis at what been done as a part of my study. We'll talk about SNPs as a predictive markers for cancer treatment. Somatically, alterations of SNPs due to lot of loss of heterozygosity and several other events are associated with the genomic instability. That's where no, we'll discuss further. Then, cancer pharmacogenomics has been explored genetic variations in candidase genes and gene pathways through critical of this physiologic process. I would like to talk about something called as cancer pharmacogenomics. For example, you might be knowing something called as xenobiotic metabolism that usually happens with the help of cytochrome 450 family of genes. They're called as something called as CYP2D6, which is an important molecule where it has to be it has to get metabolized by the CYP genes, but because of presence of an variation within the SNP, it fails to metabolize the particular drug molecules. That's where the problem arises. Now, therapeutic index for chemotherapy agents. I'll be talking shortly about the therapeutic agents. Identify genetic variants that contribute to susceptibility to cytotoxic effects. That's what we're talking about the chemotherapeutic drugs, which provide important tools to tailor therapies and decrease adverse events. The same CYP2D6 can be taken in here into consideration how it is going to influence. Now, genomic knowledge emerging from Human Genome Project and International HapMap project. There's a lot of variations that we see from Indian context, a lot of diversity that exists with Indian and with regard to taking a snip into consideration. Mm, so it allows the investigators to interrogate large set of SNPs. For example, just think for a while, what is the difference between a SNP and a mutation? What is the difference between a polymorphism and a mutation? Can anyone in the class can just throw a light on this? What is the difference between a polymorphism and a mutation?
So a variation which is present in more than 0.5% of the population will be a mutation, will be a polymorphism, and anything less, which is rare, will be,
Yeah.
or we rather use it by the minor allele frequency, which is.
Perfect, perfect. Thank you, Anshu ma'am. Thank you so much. You made the things clear. So everyone knows it. Mutation is a very rare event. That's where it happens. And in a polymorphism, for example, everyone who is listening to this, everyone has got polymorphisms, right? It's only like uh, all the genes that are present in your body, they are functioning normally. If there any tumor suppressor genes, if any oncogenes they start expressing, that's where the problem arises. This is a small understanding that I think everyone knows it. Now, as a part of this, I've taken a case study to give more brief into these kind of things. Um, when we talk something on background on leukemia, just talking about some leukemia taken into consideration, where I'm talking about both ALL and AML into consideration. I'll be talking about acute leukemia. This is part of my PhD and postdoc work, which I'm trying to discuss here. Now, we say leukemia is nothing but a group of heterogeneous hematological malignancies, which is formed because of the chromosomal abnormalities. The most important striking feature in with regard to AML and ALL is nothing but the immature cells, which are called as blast cells. This plays a pivotal role with regard to acute leukemia. If someone, sorry, these particular blast cells are very crucial in the percentage of blast cells will and define that. Basically, they're present in the bone marrow and the blood. It's all about the immature, means they are not able to form properly. That's where the problem arises. Now, absence of mature white blood cells, it leads to anemic condition. It leads to thrombocytopenia. Thrombocytopenia is with regarding to where there's a decrease um, platelet counts, and then you got leukocytosis. There is abnormal white blood cells that leads to leukocytosis. So these are the important features of a leukemic state. Now, the genetic abnormalities include dysregulated DNA repair system, failure of cell cycle checkpoints, failure of apoptosis, failure of activation of oncogenes. I told you, tumor suppressor something, but the loss of function mutations, because they're losing their function. That's where the because tumor suppressors help. For example, P53, or you talk about retinoblastoma, you talk about P21, you talk about P10. Most of them are tumor suppressor genes. If the tumor suppressor gene is losing its function, it is nothing but there is an gain of function of gain of function mutation. That's nothing but the oncogenic mutations are obvious. So loss of function mutations of tumor suppressor genes and gain of function or activation of oncogenic mutations that comes into picture. Now, then the clinical symptoms includes fatigue, shortness of breath, again exertion, easy bruising, patache, increased susceptibility. Petacha is nothing but reddening of a small bubbles, a redding bubbles will be seen on the skin. That's called as a petacha. And based upon the classification, based upon the abrupt onset and also the immature cell forms, the leukemia classification is been into acute and chronic myeloid leukemia. Chronic is nothing but it's a gradually onset of disease, severe symptoms where CML is one chronic myeloid leukemia is one disease that is you see with regard to CLL and CML when compared to acute. Acute is very abrupt, and this further divided into AML and AL and CML and CML based upon its state of cell. When I talk about leukemic epidemiology, will take into consideration in the worldwide, the cases still growing. It was around 52,318 cases. You can see uh, the different kind of mm, so stages at which, like for example, AML, you are able to see 36%, and see percentages, there varying percentages that you'll be able to see. When you see in as per the tumor history of Nizams of Medical Sciences in Hyderabad, where I was when working on, you could able to see that around 20% are belongs to ALL, then there is a 16% into AML cases. AL and AML constitute 36% of the cases what we used to get there. Now, the risk factors in acute leukemia, most important thing is that smoking, 20-25% of AML cases are linked, and chemical exposure, benzene oil refineries, chemical plants, cancer treatment, alkylating agents, platinum agents, radiation exposure, then certain blood disorders are Fanconi's anemia, and males are more prone than females. For example, I'll just give one best example. Uh, when I'm, because I basically work with clinicians, the medical oncologist. Uh, I used to ask them, like, what could be the reasons why most of the time we see agriculture coolies will be getting agriculture coolies who work in agriculture, they'll be getting mostly they come with this particular disease. I asked, like, what could be the reason, doctor? He said that, Sugunakar, it so happens that when you see the agriculture, they do a lot of pesticides, and when they spray the pesticides, and that particular pesticide will when it is sprayed, it reaches the belt, it reaches the belt beneath the soil, and it go and mix with the water, and nearby places they have lakes, they go get that water, and they don't heat them properly, or they don't use proper filtration mechanism, and they just drink it. And that's where the pesticides are one of the most important threat that currently which is there across India. Like, a lot of reports have been, we've seen a lot of reports coming down. So that is one reason that exposure, chemical exposure, or even the treatment modalities which have been there. The standard protocol that basically follows is induction, post remission therapy, a diagnosis. So what kind of analysis they do? So induction with 7+3 regimen, cytogen arabinoside, and they do it on different intervals of time. Then they use daunorubicin, and based upon kind of mutation I was telling about. For example, FLT3 is there. Now, till 2016, there was no FDA approved drug, and after that, the drug has come into picture. It is called as midostaurin and glitatinib. There are two combinations which has come. Basically, they are tyrosine kinase inhibitors that people basically go. And even in abroad, for example, in developed countries, they also go for autologous and autologous stem cell transplantation. Mm. So bone marrow transplantation, allogenic, and as well as autologous stem cell transplantation. Now, when we try and talk about the hypothesis, for example, how do many of the PhD students who are here, they might be knowing, like, how do you develop a hypothesis? How do you come out with some idea? Now, we know that we talk about when we talk about leukemia, nothing but hematopoietic stem cells. They're the lifetime survivors which are prone to damage by exposure to various exogenous and endogenous factors. And when you see this particular ATM, which is an DNA damage sensor, um, it usually initially sense the damage and activates the downstream signaling molecules. For example, the first pathway that is getting activated is NHJ pathway, and it might lead to because uh, it is activating the NHJ pathway. NHJ pathway is an error-prone pathway. I think most of you know it. Someone who has studied, they would have complete idea. It is a ligand because it's an error-prone pathway. It is ligand independent pathway. You don't have like a homologous and joining pathway. Okay, so that's where the error-prone pathway is dysregulated, and it might leads to genomic instability and also leukemogenesis. The imbalance between the proliferation, what we talk about, the proliferation that what we see in the in this, the proliferation and the apoptosis, which is associated with differentiation arrest, means if it is not able to differentiate properly into different kind of cells, that's where the real problem that exists. And that's where it undergoes survival. Then there's something called as clonal selection will come into picture. The survival in the clonal selection means the leukemic cells start growing. Okay, it's undergo proliferation. That's where it's get transformed and they undergo propagation. So which leads to leukemia. So as a part of preliminary study, I was working with Prash in the initial days. I'm talking about 2014, 2012, 2013, or from 14. We try to see that how this whatever pathway I've taken, whether it is really working properly, other than interacting with each other. That's where we try to do analysis by using string database, and we have revealed interactions with NHCJ, ATM, FLT3, FAS proteins. You can able to clearly see CBP alpha is part of an oncogenic mutation, FLT3, then able to see MAPK1 cell cycle checkpoint, then ATM gene XRCC, it's part of DNA repair. XRC7 one part of DNA repair, and XRC6 a part of DNA repair. So that's where we're able to see the interactome within this. Then we have taken various objectives with regard to epidemiological parameters, clinical parameters, for example, baseline characteristics of WBC count, platelet count, blast percentages, hemoglobin percentages, LDH levels, then complete remission and disease-free survival. I'll talk about these two things when I come to the particular slide. Then we have studied on different polymorphism on ATM, on NHJ pathway and extrinsic apoptotic pathway. In my lab, we are working on one is working on an intrinsic apoptotic pathway. Was working on extrinsic pathway. So that's how things are there. That's the things work in India. So you do on one pathway, we do on another pathway. So that's how we contribute. So then we screen for FLT3 ITD mutation, which I told you, one of the most important oncogenic mutations. That's where it's the first of our lab in India that where we try to give information to the oncologist saying that we are finding mutation in this particular patient. So the treatment modalities is changed. The treatment modality is changed. Apart from the 7+3 regimen, they also add retinoic acid, and then they used to give to the patient. So that's how. Then we did one of the objectives gene interaction studies by using LD, MDR, and combined genotype analysis. Then in silico analysis to do the study. Then as a part of study, the sources were taken from MNJ Cancer Hospital and the Ministry of Medical Sciences, Hyderabad. The total sample is around 772 samples. If you wanted to confirm any polymorphism, any SNP, it should be more than 220 samples. Okay, if you people say then the power of the sample need to be calculated, it should if you wanted to get accurate results, if you wanted to confine that this SNP is playing an important role, it should be more than 220 225 samples, but not less than 200. You cannot confine. You cannot say that you're saying that this SNP is confirmed from their end. No, it cannot be, because that is how it is. It has to be. So to confirm that this particular SNP is showing effect, so you need to work on that. So we taken acute leukemia where I have taken for it's a mostly 4.5 4 years study where every day we used to go to the cancer hospital, talk to the clinician, talk to the patient, to take his consent, and ethics committee. Everything is perfectly taken, and then we worked on these things. So it is purely on a clinical front where I used to go every day, and then we used to work on that. yeah, it takes time, but a lot of patience is required when you want to do something better for your career and also for the society. So I was thankful enough to my professor and also medical oncologist who try to help me in these things. So we have an inclusion criteria, primary de novo AML and ALL cases were taken with confirmed diagnosis based on CBP, bone marrow aspiration, and cytogenetic analysis. The exclusion criteria includes we have eliminated therapy-related and also secondary AML because of someone has got ALL and he develops into AML because of some Down syndromes and those kind of stuff. We have eliminated completely. It's a de novo, means fresh samples, fresh patients who are coming with this particular AML taken into the study. So that's how we have even excluded the criteria. The ethics committee approval will be taken from MNJ and Institute of Medical Sciences, and also our Biomedical Research Society, Institutional Ethics Committee Society, that is from USMA City, and epidemiological and clinical parameters being taken in a pro forma, and we obtained the help of the medical oncologist. This is a nutshell of methodology. Even real-time PCR is also be there part of this. So as I want to finish my PhD, so I just included only some part, even some part still there, and it's awaiting for the publications to come down. Yeah. Now we try to do results and discussion. Then we can DNA repair pathway, extrinsic apoptotic path, then clinical parameters, submolecular analysis, and FLT3 ITD. Then epidemiological variables. You can able to see that there is AML and ALL. AML is an elderly age disease. AML is an younger age disease. Means the difference would be people who have crossed 55 years of age. You try to see with AML, but it's a very rare instances you also see with the AML. To my surprise, I've seen a baby of 1.5 years who was with the AML. So it's very sad part of it. So there are some rare instances that we see in the hospitals. I was seeing it in that sample at MNJ Cancer Hospital, which is a regional cancer center in Hyderabad, which is in Red Hills. So we happen to see those rare things too. So the cytogenetics part was carrying out with uh, Lakshmi Rao at CCMB, Center for Cellular and Molecular Biology, Hyderabad, and also the sequencing part we are trying to cover with the Tangaraj group at CCMB in Hyderabad. So it's all about working on those things. Yeah. So when we try and see clearly that you're able to see that uh, within the age 20 to 30 years you see that 34.2, then more than 30 years you see 44.9, and with regarding to ALL cases it clearly says that less than 10 years you're able to see 30.8, then less than 10 to 20 years in between 42.1. And when you see with regard to the preponderance of disease, it is seen more in males when compared to the females, and the people who are living in the rural area are more prone with regard to AML and with regard to ALL. And even habitats, the smoking has seen more cases, and smokers has got seen. And there is no consanguinity because it's a somatic mutation. You don't see any consanguinity which is coming from from the parents. It's a somatic mutation, it's a sporadic mutation. That need to be clear from everyone point of view. Now, when you talk about baseline clinical characteristics and diagnosis, we take the clinical variables, that what kind of clinical variables that one need to take. Age of diagnosis, we can able to see the mean and SD standard deviation, 30.8. It is the age. Then HB levels were not that significant when compared to that. Then peripheral blast, 63.09. If it is less than, if you're getting a 5% of blast cells, you are with some abnormality, and it is you are able to see 63.3 with regarding to AML, and with regard to AL 61.1. Then WBC counts, the normal WBC counts should be between 9,000 to 14,000. But you can able to see it's going beyond 52,000. These are the important parts that we need to understand. Even the platelet count, the platelet count should be usually less than should be around 1.5 lakhs to 4.5 lakhs in a healthy individual. If it is getting down, it means that they're prone to some infections. We taken these different parameters and blast with the WBC count and platelet count, with the LDH, and also uh, other parameters. We conclude that the clinical characteristics of diagnosis has to be considered. Then there's something called as disease-free survival. What is disease-free survival? The stage at which there are no symptoms of the disease that is being observed. Okay. After chemotherapy is given, we're able to see the only 10 months of time with regarding to AML and with regard to ALL, 22.52, because as I said, acute, it's a very abrupt and it's a very progressive and highly proliferating cancer. If you take what is the percentage of people who uh, take life, people who suffered with AML and ALL. ALL is much improved when compared to AML. For example, taking a consideration of 10 patients into consideration, or with AML, out of which hardly 2 people will survive and remaining 8 people will, yeah, the, they lose their lives. So uh, that's how. But AL is much ahead of AML when compared to that. Now with the advance of FLT3 ITD inhibitors, what I was talking about, the tyrosine kinase inhibitor, it has now improved the survival rate. That's what we call as disease-free survival. Uh, and there is something called as a remission, where there is no symptoms of any disease condition. It means you're taking chemotherapy and you see no symptoms of the disease. That's called a remission state. In the remission state only, we calculate this particular disease-free survival. There's something called as relapse. Relapse is a condition where the patient who has taken chemotherapy underwent remission, again because of mutations, he again underwent relapse. Means again he developed the disease state. It's called a relapse state. And we taken we taken different SNPs into consideration. This is ATM and NHJ pathway with genes, SNPs and FLT3. These are extrinsic apoptotic pathways genes taken into consideration. Now, when we try and see with regarding to genotype and the control population, this is a case control study. Basically, we as I told you in my study design, 326 AML control cases against 221 and 226 cases. Then we try to uh, work on the models, codominant models, and the allele frequencies and the Hardwell equilibrium. And this is one SNP that we have taken, minus 5144 A to T polymorphism, means the A is changing into T in the promoter region. What exactly happens if you want to understand this? If A is changing to T, that's where the binding of the transcription factor will be changing there. That I'll show in the in silico analysis what we have done. The location is minus 44, minus 514 for position in the promoter region. It might interfere with the ATM signaling due to the loss of SP1 transcriptional factor binding site, because of changing from one single nucleotide polymorphism is going to influence a lot. So that's where we talk about SNPs, which we talk about variations with regard into the analysis. What they do, we report by using NGS the variations are what we talk about. We always talk about the SNPs which are responsible, and it has got its own impact on the disease progression. And then we try to compare with the HapMap data, which is available in other populations and in Indian population. We try to see the population. The AML and ALL controls were similar to other population, but increased in ALL. For example, we see Hispanic population, we see Japanese population, we see different populations, and we see the HapMap data. We have a HapMap data that is available. We did that work. Then we have seen the association, how it is associated. You can able to see the odds ratios here. It's clearly evident. It is showing highly significant. Now, you take different polymorphism, because different associations regard to epidemiological and clinical variables. So then we do something called as uh, survival analysis by using Kaplan-Meier curve. Um, you can able to see in the HapMap data, present study AML and present study ALL, Asian JPT, then Asian HCP, then European CEV, and Sub-Saharan African. That's where we try to calculate the HapMap data. And with this, like we were able to see that with regard to the genotypes here, AA genotype, AT genotype, and TT genotype, the variant allele AT genotype that has been clearly evidently seen that the reduced DNA repair capacity of T2 genotype and T allele may be responsible for the association in the development of the this particular thing. Then we talked about this is one SNP, ATM SNP, and this is regard to XRCC5 VNTR polymorphism, and this location is -201 in promoter region. Sorry. And then we have taken different combinations of SNPs. Because of brevity of time, I'm just trying to move a bit faster. So then we see these kind of things. Then we did the Kaplan-Meier analysis to understand presence of low repeats allele indicated excessive error error prone NHG repair, which is associated with the overexpression of XRCC5. 5. It might be result in the accumulation of somatic mutations. Lower the repeats, the error prone error is more, which is associated with the accumulation and the origin of leukemias. And we have taken XRCC 52048, which is G2A polymorphism. And then it's present in the intronic region. Even still, it is also shown some kind of evidences that it's significantly elevated, both in cases and also in the controls of both ALL and AML. And that's where I told about something called as complete remission. The patients with a variant allele have higher chance of achieving complete remission due to reduced DNA repair capacity, leading to chemosensitivity. But these patients are at higher risk for relapse because of this particular TT mutation. You need to understand the genotypes here. That's where you get a clarity on that. It's all about the genotypes. And further, when we try to do this, we're trying to establish whether we need to calculate statistically. We have to show a significance. That's where we do haplotype analysis. Haplotype is a phenomena when you see the variants in the parents, which are present in the same chromosome in the other chromosome. So we do haplotype analysis. So we are taking only the risk alleles, 2RG, 2RA, and we're taking different combinations of the genes in both XRCC5, VNTR polymorphism, and 24GA polymorphism. So it is clearly shown that the frequency of 2RA1 to IRA1, IORA have significantly elevated in both AML and ALL. Means the particular genotype, the variant allele, what we discuss, it is conferring risk irrespective of the presence of VNTR allele or not. Means the genotype, the variant genotype is showing highly risk, and these are evidently even seen by using in silico based analysis. Any variant you want to see the likely pathogenic or pathogenic in nature, you basically use shift and polyfin tools to see its impact. Now, in the same way, we did XRCC6 gene into -61 C2G polymorphism, then 1310 except with regard to XRCC6 gene. We have seen a lot of association with regard to the genotypes that is taken into consideration. So further, we have done the haplotype analysis and then diplotype analysis. What are the haplotype risk genotypes are there? We have taken those risk genotypes into the diplotype analysis, and we have seen that diplotype analysis revealed that there is an increased frequency of diplotype with 50% probability of producing risk haplotypes compared to diplotype. So there is always a higher rate when we went to diplotype analysis. Means it's a combination of the risk genotypes taken from the haplotype analysis. And this is one more SNP that we have taken. Then we have taken all the combined genotypes. For example, you have seen at least 4 to 5 SNPs till now. We have taken all those genotypes, variant alleles. Then we try to calculate with regard to the cases and controls, and we see that there is a linear increase. If you get able to see 2.28, 2.87, 7.54, means the linear there is a linear increase in odds ratio with respect to the risk genotypes in AML, and the presence of 4 risk genotypes has conferred 7.5-fold increase risk of AML. So this is an important part of statistical analysis that what we performed. Then we have taken anti-apoptotic pathway, apoptotic pathway into extrinsic apoptotic pathway, where we have taken minus 678 G polymorphism. And we have seen a lot of significant with regarding to the transcription factor binding site, then the way the genotype has contributed in the development of the disease. It's clearly evident that these risk genotypes are, whenever you see in the patients, in normal population, you don't, you see, for example, I told you I have taken 226 samples and 221 samples. In controls, you can ask me, sir, have you checked in controls? The SNPs are available. Yes, there are SNPs available, but not at the rate at which the cases are present. If the AML person is present, if all the SNPs are present as you see in the patients, it is an alarming that yes, that particular individual has to be taken proper care with regarding to his conditions is prevailing around him. So this is a FAS receptor gene. Then we have done again the haplotype analysis and the diplotype analysis. We could able to establish the study in a more better way. And then this is a FAS ligand, one more mutation we've taken. Then we've taken caspase 8. Because of brevity of time, I'm just trying to move, but only I wanted to show is like how significant the values are here. So then we did again the caspase, and we have taken 2 polymorphisms in caspases. We have done that. Then caspase 3 we have taken, and caspase 7 we see some protective nature in those. Then we have taken cumulative effect of adverse genotype of extrinsic apoptotic pathway, all the extrinsic things. Then we see, you can able to see did also shows on the p values are quite significant, which clearly says that the risk genotypes are playing a very critical role in the development of the disease. And this is an FLT3-ATD mutation, and that's what I was telling you this. And this is where a normal FLT3-ATD signaling is it's where the ligands bind onto it. But FLT3-ATD, even though there is no receptor binding, it's undergo constitutive activation. It is ligand independent dimerization, where it undergo constitutive activation. And there are some other FLT3 point mutations are also been there. Internal tandem duplication. Okay. Then you have point mutations. Then we have seen the samples that are taken into consideration. Yeah. So we calculated the disease-free survival rates, and these are the gel images. You can able to see one person has got only one internal tandem duplication. One person has got 2 internal tandem duplication, means he's more prone, and the production of the particular protein is very high. Two, again, you can able to see three bands, and you can able to see there's a varying amount of patients. We got around 34 samples, I think. Around 17% we got about this particular ITD segment. ITD segment through bioanalyzer software used by using 41 AML patients have FLT3 mutations, and 10 variants are observed in terms of sizes, different sizes that has been observed. And it is being, the normal size is being 133 base pairs. The cases range from 150 to 350 base pairs. The normal is being 133. This is a normal one. But when you see, you try to see different kind of base pair tandem duplications. When there's a duplication of the same thing. And then we have did some Cox regression analysis with regard to the FLT positive and FLT negative cases. And then we try to compare with FLT3 positive and negative with regard to age diagnosis, HBV and peripheral blast, WBC counts, platelets, and then with regard to LDL levels, we could able to see it highly significant values. Then we combined all the FLT3 ATD, then we taken all the SNPs which are available in different pathways. Then we did the statistical analysis, how exactly they are seeing with regard to FLT3 ATD. We're able to see excessive VNTR, then GTA polymorphism, caspase and caspase 7, caspase 3, and FSR. They're showing risk prediction in that. And we did this analysis, and it's part of my 2010 paper, I think, which we got in one publication. Then this is regarding to Koch regression analysis, hazardous risk ratio. So unless until you you see you show it statistically significant, your work what has done, it is of no use at all. So we did cross regression analysis, taking DFS, disease free survival, as one event, and taken hazard risk as one indicant, and then more than one which indicates risk. Okay, so it's less than one indicates the protection and more than one indicates in the risk, and we're able to see that 2408 has shown some significance, and then FSR has shown some significance. The same thing with other samples. So that's how we try to do it. Then we did gene-gene interaction, how the genes are interacting with each other by using linkage disequilibrium, multi-dimensional reduction analysis, and then combined genotype analysis. All the genotypes are combined, the risk genotypes are combined, then we did analysis. Now, LD, we did linkage disequilibrium. We see none of them are falling in the place, because they're not conserved within one chromosome on the same chromosome. They may be on the different chromosomes, but we see that with regard into the genotypes what we taken, none of them showing association, means they're showing risk independently. Okay, there is something called as epistatic. One suppresses one gene suppresses expression of the other gene. That's where we're able to see that there's an epistatic regulation that is being seen in these particular things. Okay, so this is one important thing that we have understood, means the risk is conferred independently. It is not dependent, it is independent of risk predictions. The same thing with regard into AL cases also. Then further we did MDR, multi-dimensional reduction analysis, where do iterations under different combinations, locus number and combinations. Locus 1 only has 1377 G2A, then locus 2 we have taken 2 SNPs, locus 3 we have taken 3 SNPs, locus 4, locus 5. So we did this kind of iterations by using statistics to see how the impact is there. So we use C value consistency, 8 by 10, 10 by 10, 6 by 10, and 10 by 10. We are able to see the calculate the p values, and then we calculated the dendrograms, and we are able to see that analysis revealed synergistic observed with regard to XRCC6 gene minus 61 C2G and 1370 1310 C2G. May possibly epistatic nature has been observed in haplotype analysis. Means presence of one genotype is influencing the other genotype, means it's showing, it is suppressing the effect of the other gene. That's where the epistatic images come into picture. uh, so we were able to say that the variant SNPs individually might have been associated with an aberrant NHEJ activity. Now further we did with regard to FAS gene, then we did with regard to uh, cumulative analysis, and then I started doing working on in silico based analysis and protein binding and transcriptional binding sites, intronic human splice finder, then exonic we do structure predictions by mRNA, then human splice finder, then secondary structure and tertiary structure. Then we worked on these things, we got um, results, and this is where we're going to wild type and also the mutant alleles, how it is going to form, and the genotype and phenotype expression also been studied, whether in the promoter region with regarding to how it is which is showing significance, means you can be able to see the from the functional significance. You can see the reduced DNA repair capacity, enhanced DNA repair capacity. It all depends upon the number of the repeats. Each and every SNP has got associated with some functional significance, and then we related with regard into the CR, means complete remission under disease-free survival condition, both in AML and ALL cases. So that's where you come to conclusions, unless until you will not have an idea like what is a drug that is being given, how it is going to be working. So we need to understand that. So this is possible only with the help of the medical oncologist. Now then the significance of the study, how it is being done, means if we talk about double DNA repair, if taken into consideration, then if the double strand breaks are there due to chromosomal translocations, ionizing radiations, and drugs which are part of it. So low expression and high expression of DNA repair pathways also help in cells with residual DNA damage. And there are many, that's what I tell you, there are many confounding factors which helps in the disease development. So that is not one factor, there are multiple factors that come along. No, so the significant contributions out of study that recommends that mandatory screening of FLT3 mutations in all the clinical management of AML patients. Then then FAS-L gene can be an alternative therapeutic target, and then ATM XRCC5, XRCC6 can be used as a biomarker in the management of leukemias. Now, I'm basically from LPU, and we connected last year, before last year, I think, yeah, Indian Science Congress. And I always believe in one thing: the more I study, the more I learned and observed, the more I realize how truly little I know. It was given by Steve Marabola. I'm great follower of him, so I thought I like this one. Yeah, so these are my collaborators, and yeah, my professor, then the medical oncologist who are part of this study, and then Prashant, who is my I can say a mentor, a teacher, a friend, a brother, and I see all those things in Prashant. And my lab mate Naishara Dunna is a Shastra University, and Shailaja Kagita, who are part of my journey now, and Smita Sipawar, who is at Department of Genetics, Usman City. Currently, she's the head, and one of my PhD students, Santosh Dupala, she's working with him. We're working with her on cervical cancer, and currently 7 PhDs are working with me, and then my MTech students and MScs and BTech students are part of our study. Not part of study; they're working on different projects. And yeah, thank you. So the forum is open to questions now. Yeah, please. Any questions from anyone?
Hello.
There are two things. One is you understood, other one is you have not understood. So I just wanted to know that. Yeah, please go ahead.
So actually, I have a question. You are talking about pesticides, which are released from the field and in the water bodies.
Yes, yes.
Actually, the pesticide is causing the leukemia. It's okay, but is there any special effect to children or any difference between man, woman, or children? Anything else means the leukemia, especially you are talking about that males are men means mostly affected.
Okay.
So, is there any specific effect?
So it's simple when you just use logic. Males are more work in agriculture fields than females and children. So and that's where they get exposed to pesticides. So they don't take proper precaution in handling that. That's the only thing, because for example, best example I can see, we talk about radiations, leukemia. I talk about Hiroshima, Nagasaki, the atom bomb. Most of the people there, they suffered with acute leukemia, with AML and ALL, or even irrespective of male and female there. But because it's all depend upon the kind of exposure, the amount of exposure that they are with. Any more questions? I'll be very happy to.
Yeah, wonderful talk, Sagu. Thank you so much. It was a wonderful and illustrious talk. Friends, please keep asking questions.
You can always ask in the chat, in the WhatsApp. I'm always there. Yeah.
Yeah. Now Sagu will probably give demo during the next 30 minutes or 25-30 minutes. Yeah. Please, Sagu. Thank you. Yeah.
Yeah. Thank you.
Sagu, I'm continuing the recording. Okay. Yeah.
Yeah. Fine. Gosh, no problem.
Yeah.
I'm sorry. Sorry, I switched off my mic. How many of you have an idea about Cosmic Database? How many have worked to understand the somatic mutations? Someone wanted to know about You can just type in the chat box, or you can unmute and then you can talk. I think my student Santoshi has worked a bit on Cosmic. Yeah, please. I want to listen to your replies. Uh, Anyone? No experience. Okay. Okay. Thank you, Anshu ma'am. Yeah. Okay. 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 Uh, that's fine. I'll just try to give you the link. Yeah, like I want everyone just to open, and then it's a small kind of exercise you can always do. You can select a gene of interest, then you try to understand like how it can be done. But before that, I'll just try to give you some kind of mm, uh, like how you go ahead and how you search that. I'll just try to give some kind of information. Before that, I'll just try to show some slide uh, on Cosmic just for 5 minutes quickly. Then we'll go to the website and then we start searching that. Okay, I'm just sharing my screen for 2 minutes, for 5 minutes maximum. Okay, yeah, yeah. Basically, as a part of the cancer study that God we do, we use germline and then we use somatic mutations. And the catalog of somatic mutations in cancer is all about Cosmic, which is developed by UK. And the current version is 94. It is released on 28 May 2021. And every year they try to release at least 2 to 3 times they update website with a new set of genes, which show the effects on somatic mutations across the range of all the human cancers. It is not about one, it is about all the kind of cancers. Please make sure this particular point. And it is the largest source of manually curated somatic mutation information, which is related to human cancers. And it comprises of somatic databases, and the Selenium projects are also been part of this particular study in this particular database. Okay, there are different tools that Cosmic use to explore. Nothing but Genome Browser, then Gene Pages, then cancer browsers, then fusion genes can be seen, then drug resistance data, then hallmarks of cancer, of the particular gene that you wanted to study, then Cosmic 3D structure, what are the structural variations that what you come across, then the cancer gene consensus can be studied, then cannot be studied, then cancer mutations, then uh, and currently they're trying to use GRCC 37 and 38. Both are being available. Then there are the details can be for example the paper when they these are all are I told you manually curated database. Okay, so they take all the papers taken into consideration. They do a lot of curation, and then they take the paper and they start thinking about the gene point of view, the patient point of view, and the transcripts that what is being taken. For example, age, sex, ethnicity, environmental variables, family history, and other therapies which have been taken into consideration. Further, they understand the tumor features. Regard to cytogenetics, the cytogenetics its grade, which grade of cancer it is, then drug response, then metastatic site, then further they use the mutations, which is a lot of heterozygosity or somatic status, or the zygosity or the normal tissues tested and resistant mutations taken. They work on those particular samples. They analyze by using phantom predictions and scores, Z scores, SNPs. They subject it to therapy relationship with regard to whole exome sequencing, RNA transcriptomics, and those kind of stuff. All like mutation allele specifications, like microsatellite instability has also been studied within the samples that they take into consideration. Then they see classification, the primary sites, subsites, and the histological conditions. I'll show you in a brief. Now, as per 2008 August, 18 August, there are 13 lakhs of tumor samples have been studied. For example, 59 lakhs of coding mutations have been curated. Means these are all the curated information that is available in the Cosmic database, or around 26,256 manually created publications. Then they were able to they were able to uh, keep in the database around 19,368 gene fusions have already been reported. So 30,480 whole exomes across 457 studies, and then you see copy number variations, then you see gene expression variants, or it can be methylated, and then non-coding variants. A lot of data has been explored, and it has been present in the Cosmic database. This is how basically you try and see the Cosmic database. Then you'll come to know. In the meanwhile, then you see the gene view, and these are the things that various things that you come across in the tissue distribution. Then Genome Browser, then you see the mutation distribution, how it is formed, what kind of substitutions are there, what kind of mutations type are which are available, and what are the percentages at which they're present, insertion deletions. Then you wanted to find out about those kind of things, mutations, fusions, SNV expressions, methylations, all kind of data is available, and you wanted to know which paper they've taken this particular information. You also have the references, and you can also export to a TSVs or CSV or TSV file formats, and so on and so forth. You can do that. Yeah, I'll stop sharing that. Then I'll go to website. I think most of you already opened it. Now, this is how a website looks like. The most recent one I told you, it's a version 94 released on 28 May 21. Then you try to see, and you can able to see on projects, you see like a Cosmic Cell and projects, Cosmic 3D, Cosmic Gene Consensus, Census, Cancer Gene,
So we are not
Cancer
able to
Mutation.
see your screen.
Oh, I'm
Your
sorry.
screen is not visible.
I'm sorry, I did not share it. Yeah, yeah. Now you're able to see it?
Yes, sir. Now it is visible.
Oh, I'm sorry. Yeah, now this is a Cosmic database that basically you see, like when you see projects, you see Cosmic Cell and Study projects, then Cosmic 3D, then Cancer Gene Consensus. You have downloads. You can always download by using a SFFT site. You can download all the things that what exactly you want. You can always do that. And then you have a datas and datas which are going to gene curation, gene fusion curation, genome screens, and then drug resistance, mutational. You can see a lot of data. Before that, I just wanted to start with, for example, you wanted to know how, for example, yeah, it's only demo. I want you to work on some gene of interest, and then you can ask me any question that you get as a part of the next discussion till 19th or 20th, or even after that. You can always contact me or Prash by using the email address that is what has been given. If you have any further doubts, you can always go to the publications. For example, when you click on the publications, you can able to clearly see there are 10 publications just come from the year 2004. The first work started with 4 genes. The first curation started with the manual curation started with the 4 genes in 2004, and we have seen now how many genes which has coming now, which has come now. Okay, so you can see all the data that it's all. You can see the release notes, how many genes have been come, the data updates. You can able to see now version 28 on 28th of May 2021. They have released the curated genes, and every year this year they have released two, May and March, and in 2020 you can see August, April, and August, April. Because of pandemic, I think it has come down. Otherwise, they at least release 3 to 4 releases per year. Um, so into the 19 you can able to see. So you see a lot of data that is available uh, 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 about this. Now, when we try and go here, there is something called as, which are, you see something called tools. Okay, and someone wanted to, for example, Anshumam she's a pathologist and she wanted to work on something. Anshumam, are you around? She might be.
Yes, sir. I'm there.
Yeah, she might be interested in knowing the tissue type and the histology type. If she can just go to this particular genome browser, cancer browser, and she can do the tissue selection, sub-tissue selection, histology selection, and then sub-histology selection. It's all about like what you select and how you go ahead. For example, taking cervix into consideration, and then you include whether you wanted to include it, you don't want to include it, then for example, for example, just want to include that, and sub tissue selection, you include all. Then in histology section, I wanted to look at only the carcinoma part. Then in sub histology part, I wanted to again further select only the squamous cell carcinoma, right? And this is how you do selection with regarding to each and every type. You can take any cancer into consideration. It's not about cervical, it's not about leukemia, it's about any skin, any kind of study that you take into consideration, ovarian, esophagus, pancreas, then liver, kidney, hematological lymph nodes. Then it is your interest that you can go. Then once select, just go. Then once you get this, you try to get the list of top 20 genes, the genes with mutations, the genes without mutations. You can able to see those stuff, and genes with mutations, you can able to see the genes which are associated with this particular cervical. I'm able to see the list of all those. And if I want to include all the genes in the plot, I can also include those kind of things. And genes with mutations, you can able to see like 409 4955. You can see the all the entries which are being there, means it's completely manually curated. You wanted to know its details, each and every gene that you taking into consideration, you can always go. And then you can go to the this is with regard to the genes list, that what you get it. And you can always export with regard to your CSV and TSV files, and then you do your the other analysis. Okay, they can do, you can look at the clinical where, as Prashant was telling about DBSNPs. You can look at the SNPs, and you can also you'll see now when you're coming down, you can always use the Genome Browser. And here we have to use different kind of filters. Means you wanted to know the ensemble transcripts, the non-coding RNAs, and the regulatory features you wanted to know, the micro RNA you want to know. Then you want to see the copy number variations, deletions, gene expressions. See, it's all about that. You need to search it, and then get to know about it, and then select what exactly you wanted to really work on. Uh, then you have it taking some time. You see mutation matrix, then the distribution, and you can always filter out like what exactly it is clearly shown here. Your selection, what selection you have done. Yeah,
Can you get data geographical region wise here?
uh, that I've not seen it, ma'am. Geographical area, I've not seen that particular part. I think based upon the publications that what you get, you can always just you can get to know about through that. I seriously have not seen these geographical things. If anything I find out, certainly I'll let you know about that.
Thank you.
Yeah, yeah.
Sir, can you please explain what is being shown by those red lines in the genome browser? In the genome browser part, these red lines, what is being meant by this?
No, no, this is a complete stretch of that particular gene across the human browser. For example, you have an ensemble where you try to see all the transcripts, the complete length of the gene, and how it has been present. For example, if you want to click here, you can able to see the transcript ID and the chromosome 17 and the start site and the end site. Okay, and you can always
Okay, sir.
extend it. You can see, you can extend, you can start reading about each and everything what you are looking at, which gene you want to look at, and which position you want to look at. It's all about, only you need to spend time and then work on those genes which you wanted to look at. You wanted to know its regulatory features, you can always try to know about it. All kind of deletions, substitutions, you can get to know about it. So
So can you give me an example of maybe if I'm working in breast cancer and I want to look at a gene, so how will this data help me? It's like if I have patients of breast cancer, And what do we, how will this database help me? I'm not very clear about what to do with this.
I like, okay. Let me just give one example of one gene that is taken into consideration. Just a minute. Once I finish this, I'll just give you that example. Yeah. Uh, I want this mutation matrix. I think it is taking time to load, and you can also see the distribution uh, of this particular mutation type. Uh, this is nonsense mutations, then missense mutations across the samples that is taken into consideration. You can see frame shift mutations, and then the kind of substitution that it has got, the mutation type, how it is changing across the complete set of samples that is taken in consideration, how many are into A to C and how many are into A to G and into different combinations. And this is how you try and see about distribution of this. And then further you try to see the deletions with regarding to the samples. uh, and further you try to see the variants, the mutations that you wanted to see. For example, you have variant, you have fusion genes, you want to see the methylations, SNVs. For example, as and she's asking, like, for example, we wanted to know which genes and how do you find out. You can always get the genes information and the samples here, the kind of sample which is taken from a TCGA database. And um, uh, as I told you, it's manually curated, and you get all the data that is taken across.
So are these going to be used as a reference gene for them?
Yes, ma'am. And this is how you try and see. Uh, I think this is taking time to load. For example, I've taken an example, P10, which is a tumor suppressor gene. Okay, and I wanted to know about this. What is you get all the details of entrance ID, the genome location, which tire, and then you wanted to know what is the feature of this particular gene. You can always click here, then you can know what is the basic feature of each about this particular P10. uh, you can switch on, then you can see what kind of functions does it play. And proliferative signaling, with regard to that, then suppression of growth, basically that is what the role of this particular P10 is taken into consideration. And ischemic mimicking response to cancer, then you try and see about the nature of this particular the role in cancer. It is a TSG, and sometimes it also tumor suppressor gene. Then it also shows the impact in senescence, and it helps the role in cell division. And then which is completely connected to the PubMed. If you just click it on, you get the particular reference link, and then you can go and study about that particular thing. So you try and see that about that. And then just one sec. Just 1 minute. Yeah, this is regarding to the PTEN mutation, where we try to see about the 3D structure of the protein, then the consensus gene, the curated gene, and related to the mouse gene and the hallmarks of the gene. You wanted to know about this, you can always click on the 3D structure. Then you can able to see this information about the 3D structure of the protein, because it has got 6 structures in PTEN. You can always get to know about that. And any drug resistance with regarding to this particular PTEN can also be seen, and you can able to see the point mutations and across the tissues and with regard to different tissues. And for example, now as Anshu asked, kind of the breast cancer, if you want to know about PTEN, you can always see here. And with regard to the point mutations, the combination variations and the expressions, and those kind of information can be taken from different tissues taken into consideration. So uh, I think this data can be then be used by using CleanWire and Genome MD to see its effect. I think Prash able to guide you much better with regarding to this.
Wonderful, Sugu. I mean, this has come up with a very wonderful setting. I think, dear participants, it's time to thank Sugu for his wonderful talk. So let's, please unmute your microphone and give him a thumping round of applause, please. Yeah. So thank you, Sugu. I'll just quickly take 10 minutes. So if you have any questions, maybe Sugu can send you a Google Meet. He can give you more demonstration. But what I thought was I'll quickly share with you how these particular variants can be interpreted. I'll just quickly take 10 minutes. Okay. So what you could do is you can go to NCBI, which is a gen bank. You can quickly go to SNP here. Okay, as you could see. Can you see the screen, please? Yeah, no?
Yes, we can see your screen now.
Yeah, good. Okay, please go to SNP here in NCBI. Okay, so for example, if I give something like, say, prostate cancer, okay, you can also give organism as well. Usually prostate cancer, many strains are seen in Homo sapiens. So you can give Homo sapiens like this, like how Sunita has demonstrated, okay, and you can give in quotations. So you will get all the SNPs related to prostate cancer in human beings, okay. So if you see here, you'll get all the things. Probably you can get SNPs and DB bar and something like this. You'll also get CleanVar mutations of 37,175. CleanVar are the most precise ones. So if you don't give this, you can also try with this. Okay, these are all the CleanVar variants. Okay, so you see here at the clinical significance at the extreme left, you get, pathogenic, likely pathogenic, uncertain significance, likely benign, and benign. Benign is nothing but non-malignant. Okay, you should not consider them. Likely benign also, they have very less confidence. Risk factor you might take, but to us, the most important thing would be pathogenic variants. If you can click it, out of 37,175, you're filtering them. You can also consider likely pathogenic. They might have better significance. Okay. Uncertain significance also, they might be very much important. Okay. And again, the type of mutations, where these mutations are seen, are the frame shift, which means change in amino acid, missense, nonsense, splice site, non-coding RNAs, and neogene, okay, or UTRs, which are untranslated regions. So to me, the most important thing are nonsense mutations, okay, which might result in new proteins, okay, and also missense as well, okay. So if you take them, you see you have jotted down to 2,928 mutations out of 37,000, okay. And again, the type of mutations, of course, many of you may not really be interested in the multiple nucleotide polymorphisms. You might really look into only the SNPs, okay, the single nucleotide polymorphisms, okay. So that would come down to 2,377. And again, the variant length, usually if there are SNPs, they're usually less than 51 bases, usually one basis. That is where you are really looking into. Single submitters and all those kind of things, you would probably look into whether or not it's germline or somatic. Somatic means, which is not really, germline, which means it's not inherited. So germline is inherited mutations. And the most important thing would be to consider the method type, clinical testing, literature only, which means they're published in PubMed or whatever. Okay, you can further filter. But to me, these are okay, which means out of prostate cancer mutations in humans, you have jotted down to 2,377. I simply download them, download file. Okay, you can take tabular format. Okay, find tabular or summary or whatever. Okay, then create a file, and then your file is downloaded. And you can open it in Excel worksheet. The file size might be a bit bigger. Okay, so it's still downloading. I'll open it, for you. Don't worry. Okay. So I think it's downloaded. So I'm just opening it. So what you could do is, once you download it, you can open it with Excel. Okay, like this. So when you get it, you can open it like this with a tab delimited option, and you'll get it like this. Okay, so simply the first row is your heading, simply keep it bold. Okay, for brevity, just keep your columns like this, small columns, so that you can see other columns as well. Okay, and keep this as a filter. You're filtering it. Okay, and of course, the most important thing is you should consider the chromosome 38. Chromosome 38 is your build. It's the latest build. Okay, and DB-SNP ID and all those kind of things, RSID, RSID is your reference-seq ID, the canonical mutation. Okay, all of these things, you are getting it. So what you could do is, the reason behind this is, I know that prostate cancer is caused by BRCA mutation, which is a well-known. Okay, I can only consider only the BRCA2 and BRCA1 mutations. Okay, I can click it. So now I see I'm filtering it. I'm getting only those few mutations. But I can say that many of these conditions need not be BRCA, doesn't necessarily mean that it's only breast cancer. It could even be prostate cancer specifically. So if I can go down, I can say that it's a hereditary cancer predisposed or something. Okay, so it's pancreatic, and some of them are not related to hereditary. Okay, maybe I can go for malignant tumor like this. Okay, only consider it. You can even type malignant here. Okay, whatever you want, and click OK. And then you are getting only those variants that are specific to, say, malignant. Sorry, like this. Okay. Okay. So you see, out of 37,000, we have jotted down to 2,300. And now I can say vehemently that we have found two important genes here that are specifically protein changing, which means they're absolutely causal and they're responsible for malignancy. And now I can even go back and see if these mutations are, there with RSIDs. Yes, there are RSIDs. You can copy it. Okay. And now go to SNP database and double check it if those RSIDs are there. You can give comma and search. Okay, I don't know why it is getting like this, but you can also do like this. Go to NCBI website, go to SNP, okay, like this. You can also search one by one as well, okay. And now you see it is giving you a pathogenic list, okay. But here I don't see the minor allele frequency. Sometimes we may not really get the minor allele frequency because minor allele frequency is also very much important. But if it is pathogenic, it should be less than what you call 0.05. I'm searching the other RSID, okay. And here also I don't see the minor allele frequency, but it's likely pathogenic. But I can definitely see this. If you really want this, you can search this in something called Vure. Okay, Vure is actually a database, okay, like this, which will tell you what are the variants now specifically that are responsible for your phenotype. Simply click that, okay, and search. Sorry, it is not, maybe I can sign in. Yeah, I can sign in, otherwise you can also search like this, Vure. Okay. I don't know why. Yeah, it's asking me to sign in actually. I don't know. Yeah, 1 minute. Okay. Oh, 1 minute, please. I don't know. Yeah, I did not get my email yet. 1 minute. Okay. This is one database where you can see it. Then you can see the list of variants. You can also use Genome. Okay. Genome is also another database which will tell you the variants. Simply go for version 3.1.1, which is for HG38, and simply type your RSID. Okay. So this probably I'll type it here in your this thing. Okay. So it says that usually you should get it actually. Okay. You can also search the other SNP that we have searched. Okay. Or otherwise you can also search like this chromosome. This is GRCH38. Okay. Search like this. Sorry.
What exactly is GRCH38?
GRCH is a reference, human genome build reference, actually. Okay, that's the HG38 which we have searched, right? Yeah. So it is searching, it is giving this. So once I click this for 38, it is telling me that this is the position. Okay. So which means it's a multiple nucleotide polymorphism. It could be a copy number variant. Okay. But your variant is 32336339 in chromosome 13. So if you can come down, its 97 is somewhere here. Okay. Can you see 97 somewhere? Yeah. I don't think it's at 97 only. 32363397. Yeah. 32363397. 97 is not listed, which means it is not a good candidate, which means it is not seen actually. Maybe it is misannotated, although it's BRCA2, as you could see here. The allele count is also very less. Okay. Allele frequency, although it's good, but it is seen in some homozygotes. And although there are missense mutations, it might be benign. Okay, it's 38, as you could see. You should also see this thing as well. If it is 32363397, it is T that is replaced with A or G, which means the reference allele is T and alternative allele is A or G. Okay, so T, A or G, you don't find it here, which means it can be a false positive. Okay. So this is how you really need to interpret. First go to CleanVar or SNP, search your phenotype, get the variants, download the file, open an Excel worksheet, and then check for the proprietary databases like Genome and cross-check whether your variant is clinically pathogenic or not. Okay. So I'm sorry, we need to probably stop here. If you have any questions, just please post me in WhatsApp. Okay. So thank you for today. Please do these exercises as wonderfully told by Sugu. And if you have any questions, please let us know. Okay. Yeah. Thank you so much. So if you have any questions, Sugu can take over in Google Meet. Anyone of you have got any questions, please? Yeah. Thank you.
Yeah. Thank you. Thank you all.